REDOX webinars, which are organized together with other colleagues, namely Alessandro Gioffre from the Italian Research Council, Professor Mauro Maccarone from the University of L'Aquila, Marzia Arese from Sapienza University of Rome, Elena Forte, Sapienza University of Rome, and Alessandro Leuti from the campus Biomedico University of Rome. It is a great pleasure today to have Professor Alan Butterfield as a chair of the symposium. And Professor Alan Butterfield, of course, does not need any introduction, but just to say a few words about him, I would like to remark that he's a professor of biological chemistry at the University of Kentucky. He's also associate vice president for research, faculty of the Sanders Brown Center on Aging, and fellow of the Society for Redox Biology and Medicine at the University of Kentucky. Professor Butterfield's career is marked by his seminal research on the oxidative damage in the brains of persons with Alzheimer's disease and its earlier stage, mild cognitive impairment. He also pioneered the methods of redox proteomics to identify oxidatively modified brain proteins in persons with AD and MCI. Professor Butterfield showed that amyloid beta peptide oligomers lead to lipid peroxidation, which leads to the neurotoxic product 4-hydroxy-2-nonenal. Professor Butterfield demonstrated that this highly reactive molecule binds to and causes dysfunction of brain proteins, among which are those associated with glucose metabolism, proteostasis, insulin signaling, all of which are dysfunctional in AD and MCI. His research has resulted in more than 630 publications with an H-index of 123 and has been supported by grants from the NIH. He has graduated 68 PhD and master students and trained more than 30 postdoctoral scholars and visiting scientists. Some of them, I can add, are in Italy. So many thanks, Alan, and I give you the floor to chair this symposium.
Thank you very much, Professor Sasso. Glad to welcome everyone from all over the world to this virtual symposium on redox biology and pharmacology and disease. So we have three outstanding presenters today. Professor Barry Hallowell from the National University of Singapore, Associate Professor Eugenio Barone from Sapienza University of Rome, and Professor Dorette St. Clair at the University of Kentucky in the United States. I ask you to please use the chat function in Zoom to put your questions about any of the three presentations. And after the last presentation by Professor St. Clair, then we will have a question and answer period. If you wish, you can add your name and your institution to your questions. But in the meantime, please mute your microphones and turn off your camera. This helps both to eliminate any background noises that can be disturbing to the speakers, but also saves bandwidth, which is helpful. And of course, when we have question and answer period, if I will read the questions to and direct them to the right person. Excuse me. And if you wish to follow up or something, turn on your microphone. So I will now introduce all three speakers at the beginning here so that we can just go from one into the other, and then we'll have the Q&A afterwards. So our first presenter is the Distinguished Professor of Biochemistry at the National University of Singapore, Professor Barry Hallowell. He is widely recognized internationally as one of the pioneers in the whole field of redox biology and medicine. And his book of that same title is now in its 5th edition. Amazing. His prodigious research output is continually cited by scientists all over the world, and his H-index is arguably among the highest I've ever seen for a biochemistry professor. In fact, maybe for any discipline, way past 160 something, 169 or something like that. It's way up there. Anyway, because of the current time in Singapore is really quite late there, and due to other required tasks that he had to do, Professor Hallowell's presentation will be given by video. However, a senior member of his laboratory, Dr. Erwin Chia, will take questions about Professor Hallowell's presentation following the last presentation. And his video is entitled Adventures in Ergothioneine. Our second presentation will be given by Associate Professor of Biochemical Society at the Sapienza University of Rome, Dr. Eugenio Barone. Dr. Barone has developed an international recognition for his really outstanding seminal research on insulin signaling and insulin resistance in neurodegenerative disorders like Alzheimer's disease and Down syndrome, based in part on his publishing over 150 papers and his excellent funding from several European funding agencies. Professor Barone's presentation is entitled Insulin Signaling in Alzheimer's Disease Brain and Models Thereof. And the third presentation of this symposium on redox biology and pharmacology in disease will be given by Professor Dorette St. Clair. She is the James Graham Brown Foundation Chair in Neurosciences in the Department of Toxicology and Cancer Biology at the University of Kentucky. She also is the Associate Director of Basic Research at the National Cancer Institute designated Markey Cancer Center here at UK. Professor St. Clair is internationally acknowledged as the main discoverer of the role of manganese superoxide dismutase, or MNSOD, in cancer and the elevated redox state of cancer cells. She's also shown that redox active chemotherapy agents can cause selective death of cancer cells while protecting normal cells. Professor St. Clair has published a very large number of comprehensive scientific papers supported by her essentially continuous funding from the National Cancer Institute of NIH with multiple grants simultaneously. Professor St. Clair's presentation today at the symposium today is entitled Redox Biology of Cancer and Cancer Chemotherapy, Two Birds with One Stone. So I will now ask Professor Sasso and Marco Gori to start the video for Professor Hallowell's presentation.
Hi, good afternoon, everybody, from a rather wet and rainy Singapore. I'd like to thank the organisers for inviting me to speak at this prestigious virtual symposium on redox biology and pharmacology in disease. I'm going to speak about our work with a very interesting diet-derived compound called ergothioneine. But before I start, let me introduce a key member of my laboratory, Dr. Erwin Chia, Senior Research Fellow, who's done most of the work that I'm going to speak about, and he will be answering any questions that come from this presentation, because unfortunately, when it is played to you, I will be overseas. Now, research in my laboratory for approximately 50 years, which is a scary number, has been devoted to trying to understand the role of free radicals and antioxidants in human health and disease, particularly in neurodegenerative disease. We're also very interested in the role of free radicals and antioxidants in aging, which we study both in a simple model organism, the nematode C. elegans, and on the other aspect, and basically we study it in aging cohorts of aging humans. Well, as you all know, in the human body, we constantly make oxygen radicals and other reactive oxygen species. Some of them are made accidentally when electrons escape from electron transport chains. Others are made deliberately by NADPH oxidases and mitochondria. They play very important signaling roles, and they play very important roles in our defense against pathogens. Balancing this constant production of reactive oxygen species, we have a very complex antioxidant defense network. And that comprises antioxidants that we make ourselves in the human body, like peroxyredoxins and glutathione, superoxide dismutase and catalase, and so on. And there are a number of antioxidants, or so-called antioxidants, that we actually get from the diet. And most attention has been paid to vitamin E, vitamin C, carotenoids, and various monophenols and polyphenols, such as the flavonoids. Now, the function of the antioxidant defense network is not to eliminate all the reactive oxygen species, because they have a number of very useful roles in defense and signaling, as I mentioned earlier, but to modulate their levels so that their useful functions outweigh their ability to damage biomolecules, the so-called oxidative damage. And as a result of this, oxidative damage occurs constantly in the human body, and you either repair it, which is important in the case of DNA, or you simply replace the damaged molecules, which is what happens with RNA, with proteins, and with damaged carbohydrates. And so both antioxidant defense and also repair processes are very important in minimizing oxidative damage. And it's important to do that because oxidative damage is linked to the origin and progression of a number of human diseases. And as you tend to get older, you tend to make more reactive oxygen species. The levels of certain antioxidants, by no means all of them, but some of them, like reduced glutathione, tend to decrease, and the efficiency of the various repair systems tends to decline. Now, one particular organ that's often said to be prone to oxidative damage is, of course, the brain. It's very rich in PUFA, so it's especially prone to lipid oxidation. And in fact, it's been known for many years that if brain is damaged, iron-dependent oxidative damage, especially lipid peroxidation, occurs at a very rapid rate. And in particular, work by many groups globally, especially work from Professor Alan Butterfield's laboratory at the University of Kentucky, has shown that levels of oxidative damage are elevated in the affected brain regions in patients with Parkinson's disease. And this is a whole set of biomarkers of oxidative damage to DNA, RNA, lipids, and proteins, and they're all elevated. And this is probably due both to increased production of reactive oxygen species and to decreased clearance due to failures in the normal mechanism for clearing oxidatively damaged molecules. What is even more significant, work by Alan and many others has shown that levels of oxidative damage are already elevated if you look at the brains of people with mild cognitive impairment, or amnestic mild cognitive impairment to be specific, which implies that increased oxidative damage is an early event in the development of dementia. Thus, it may precede the development of dementia. And therefore, it would seem to be obvious that antioxidant therapies could be very useful in delaying the onset or progression of dementia. Unfortunately, the things that have been tested so far have not panned out very well. Vitamin E is a fat-soluble vitamin essential in the human diet. If you are deficient in it, you get neurodegeneration and increased lipid peroxidation. In fact, its only well-established role in the human body is an antioxidant protecting lipids. However, high-dose vitamin E has been tested in dementia patients, and it really has very little effect. There were some early positive studies, but they in fact have not been repeated. So in general, the intervention trials with high-dose vitamin E in the treatment or prevention of the development of Alzheimer's disease have really overall given quite negative results. And similar studies with vitamin C and carotenoids again have given negative results. And there's also suggestions that high doses of some of these compounds are not in fact harmless, that high doses of vitamin E may increase your risk of hemorrhagic stroke. So since we know oxidative damage is important in Alzheimer's disease and other dementias, why did giving antioxidants not work particularly well? And there are a number of reasons, but two in particular apply to neurodegenerative disease. Firstly, it's very difficult to raise brain levels. The brain has a certain level of vitamin E and a certain level of vitamin C, and if you administer more to a human subject, the brain levels do not go up very much. The other problem is that high doses don't actually seem to decrease oxidative damage any better than low doses. So the RDA of vitamin E is very effective at controlling lipid peroxidation. Doesn't control it completely, of course, as I mentioned earlier, but higher doses don't seem to be any better. And of course, if you didn't decrease oxidative damage, you would predict no effect on the disease pathology, which is exactly what you see. And that brings me to ergothioneine. Ergothioneine is a thiol-thione derivative of histidine, and its chemical structure is shown there. It spends most of its time in the thione form, in a tautomeric equilibrium. And because of that, it's actually much more stable than most thiols. It was first identified in the ergot fungus in the early 1900s, hence the name ergothioneine. Now, ergothioneine is present in the human diet, as I'll explain a little later. And when you consume it, it's absorbed very effectively into the human body. It's distributed to our tissues and it is retained. Now, a very large amount of work on ergothioneine was done in the 1950s, and there's an excellent review by Melville, a pioneer in this area, published in Vitamins and Hormones in 1959. We did work on it in the 1980s, published it in 1991, where we characterized the antioxidant action of ergothioneine. It is able to scavenge a number of reactive oxygen species. It can also bind iron and copper in non-redox active forms. And since then, a number of people have tested the effects of ergothioneine in various animal models of human disease. And it's actually, in these animal models, has shown to be very protective in some models of ARDS, endothelial dysfunction, ischemia, reperfusion, fibrosis, and so on. In general, these are all fairly old papers, although the literature is very sound. And this article here was during COVID, we couldn't actually work in the lab, so we wrote an article suggesting that ergothioneine might be useful in the treatment of COVID patients because it has all these biological effects. Now, the ergothioneine transporter that brings it into the human body and distributes it into all our tissues is called OCTN1. Ergothioneine itself cannot cross cell membranes because it's a very hydrophilic molecule. So it only goes where the ergothioneine transporter is. And if you knock out this transporter in mice, they cannot take up ergothioneine. As I mentioned earlier, when you consume ergothioneine, it's avidly taken up and retained. Now, there is also ergothioneine in cell culture media. It's present in fetal calf serum. And if the cells have the transporter, as for example, HeLa cells do, they actually take up and accumulate ergothioneine very effectively against a concentration gradient. So ergothioneine comes from the human diet. The major source in the human diet is mushrooms. Mushrooms are extremely rich in ergothioneine, but you also find it in many other foods. Recently, we studied the effect of the levels of ergothioneine in asparagus, Now, ergothioneine can only be made by fungi and certain bacteria. Animals and higher plants, as far as we know, cannot make it. Now, asparagus is a higher plant, yet some batches of asparagus contain a lot of ergothioneine and others do not. And it doesn't have the genes to make ergothioneine. And what appears to happen is that fungi in the soil can supply ergothioneine, and it's taken up by many plants. And I refer you here to the excellent papers of Bob Beelman, who studied this in great depth. Now, we became interested in ergothioneine for various reasons, and we were studying people with age, and we found, in fact, that ergothioneine levels in elderly Singaporeans decrease as you get older. After age about 70, 80, they begin to decrease. What was actually more significant is that those elderly individuals who had mild cognitive impairment had much lower decline, much lower levels of basal plasma ergothioneine. And this was our initial study. We only had 25 cognitively impaired subjects and 25 age-matched elderly controls, but you could see a very, very clear difference between the two. And at about the same time, other scientists were showing that low blood levels of ergothioneine are a risk factor for the development of Parkinson's disease, for frailty, and for cardiovascular disease. So all these papers showed that if you had a low blood level of ergothioneine, your risk of Parkinson's, of cognitive impairment, of frailty, and cardiovascular disease was actually increased. Now, then we were fortunate to work with our Memory, Aging and Cognition Centre, Chris Chen and Mitchell Lai at our National University's health system. And they study a lot of cognitively impaired and demented people. And we were able to demonstrate that the effects that we'd shown were seen when you move from non-cognitively impaired subjects to mild cognitive impairment. And when they moved on to full dementia, the plasma ergothioneine levels were decreasing. Now, this is typical human data. You'll notice that most people cluster within the boxes, and there's a significant drop when you go from NCI to cognitive impairment to full dementia. But of course, there are always outliers, and these outliers always tell you that there are interesting things going on. But in the majority of patients with cognitive impairment and dementia, ergothioneine levels are significantly decreased. Now, what was even more interesting was that the Memory, Aging, Cognition Centre has followed patients over a number of years. And what they showed was that people who had low initial ergothioneine levels, when you followed them for up to 5 years, their rate of cognitive decline was greater than in people who had a higher ergothioneine level. So in almost every index of cognitive impairment, if you started with lower initial blood levels of ergothioneine, you had faster cognitive and functional decline. So when we put all this data together and combined it with the data from other groups, we were able to say that there is a sort of minimum plasma level of ergothioneine that's associated with lower risk of age-related disease. And it's about a bit less than 200 micromolar. And if you're below that, and this is plasma level, if you're below that, your risk of vascular dementia, of Alzheimer's and mild cognitive impairment is increased. Your risk of Parkinson's disease is increased. And more recently, in work with the Singapore Eye Research Institute, we've shown that your risk of AMD, age-related macular degeneration, is also increased. So our hypothesis is that ergothioneine, low levels of ergothioneine, predispose to age-related disease, especially neurodegenerative disease. Supporting this hypothesis is the fact that a number of studies have shown that ergothioneine can be neuroprotective in isolated cells against beta amyloid toxicity, against NMDA excitotoxicity in retinal neurons, in mice protecting against damage by injecting beta amyloid into the brain, and most recently in a mouse model of Alzheimer's disease, administration of ergothioneine restored glucose metabolism and decreased levels of oxidative stress. So there's some evidence of protection. Ergothioneine is present not only in the brains of adult humans, it's also present in the brains of babies. And this is a paper describing the metabolome of brains from very young infants who die very sadly of sudden infant death syndrome. And among the metabolites identified was ergothioneine. And we have evidence that ergothioneine can cross the placenta from the mother into the baby, and it may also be delivered to the baby in the mother's milk. When the mother is pregnant, the levels of the transporter OCTN1 in the mammary gland appear to increase, and it seems to be that they're delivering ergothioneine to the baby, which again suggests that ergothioneine is extremely important to us. Also consistent with this idea of the protective role of ergothioneine is a number of studies, including one by us, that show that increased mushroom consumption is associated with a lower risk of mild cognitive impairment. And this has been shown in Japan, it's been shown in our study in Singapore, and it's been shown in the United States from the NAIN study, and that they look more broadly at all cause and cause-specific mortality. So consuming more mushrooms, again, seems to be associated with lower risk of age-related disease. Although, of course, mushrooms are rich in ergothioneine, they contain many other protective compounds as well. So just to show you some data as to how well ergothioneine is retained in the body, this is a study that Irwin and Richard Tang performed, feeding ergothioneine to mice. Now, the levels of ergothioneine in mouse chow are actually very low, yet the mice take up these low levels and they accumulate them. So we actually find that the ergothioneine content of tissues in these mice is already quite substantial. And when we feed more ergothioneine, the levels in every tissue we examined increase. Liver, heart, spleen, lung, kidney, bone marrow, the eye, and particularly important to what we're talking about today, it crossed the blood-brain barrier and it accumulated into the brain. And this shows that after 28 days, there was a very substantial increase in the ergothioneine levels in the brain. So it can cross the blood-brain barrier and it can accumulate in the brain. Now, the data that I've shown you suggest that low intakes or blood levels of ergothioneine predispose to age-related diseases. But of course, correlation does not mean causation. There is an alternative explanation that age-related diseases cause falls in ergothioneine levels, perhaps because of changes in diet, that maybe they impair intestinal absorption, that more ergothioneine is used, or maybe the kidneys are not working so well and the renal reabsorption of ergothioneine is slowed. So distinguishing cause from consequence requires direct experiments. And I've already indicated a number of these, but let me show you a few more. Here is a C. elegans model of dementia in which you overexpress beta amyloid in the muscle and the animals develop paralysis and premature death, and treating them with ergothioneine in their diets significantly and dose-dependently increased their survival and increased their activity. I've already mentioned the animal studies on the mouse model of Alzheimer's disease, where it was extremely protective. And our group and others is studying the role of ergothioneine in a great range of other diseases. So I'll just show you one with Parkinson's disease. There are various models of Parkinson's disease, but one of them is a Drosophila model. And if you knock out parkin in Drosophila, they develop a movement disorder, and this movement disorder is reversed by ergothioneine. And you can actually see that ergothioneine protects the dopaminergic neurons in these flies which are missing parkin. There are various inherited forms of Parkinson's disease, and if in our fly models we express a mutant form of alpha-synuclein or a mutant form of LRRK2, and both these mutant forms are known to be associated with development of inherited Parkinson's disease in human subjects, you again get a movement disorder in the flies, and this is protected against by giving the animals ergothioneine. Another model of Parkinson's disease in rodents is the administration of 6-hydroxydopamine, direct injection into the brain. It kills cells in the substantia nigra. Treatment with ergothioneine abolishes the movement disorder and preserves the cells in the substantia nigra. So this is an animal model of Parkinson's disease, and in both fly and animal models, ergothioneine is very protective. So the data that's being accumulated rapidly by us and many other groups suggests that ergothioneine is protective. But then the big question is it protective because of its antioxidant abilities, or is it protective because of the other abilities that I alluded to earlier? Well, the only group who studied the effects of administering ergothioneine to human subjects on biomarkers of oxidative damage is us. And we gave pure ergothioneine to healthy young human subjects. And we saw some trends in the decrease of biomarkers of oxidative damage, as mentioned here, but most of the changes were not significant. So at least in these healthy subjects, giving ergothioneine did not seem to decrease their levels of oxidative damage. But of course, there are many antioxidants already in human tissues, like reduced glutathione, superoxide dismutase, all the ones I mentioned earlier, and of course vitamin E. We have evidence from our studies and studies from many groups that when human tissues or animal tissues are injured, there is an upregulation of the message encoding OCTN1. And this has been shown in fatty liver, in damaged heart, in preeclampsia, and in retinal trauma. So OCTN1 levels increase and more ergothioneine is brought into the damaged tissue. And in the studies that have examined this, if you stop that intake of ergothioneine, the damage increases. So we suspect that in the normal human body, ergothioneine is probably not a major antioxidant, but in damaged tissues, you increase its levels and it may get pumped in with the aim of decreasing oxidative damage. But as I said earlier, ergothioneine has many other properties as well. Again, this literature is very old, but that doesn't mean it's bad literature. So ergothioneine could be protective by a number of different mechanisms. So we believe that ergothioneine is a very exciting compound. It's a compound that comes from diet. It's accumulated high levels in the human body. We retain it, it's reabsorbed in the kidneys. It's converted or it's transmitted from mother to baby. So it obviously seems to be very important. So at the end, I'd like to thank the members of my research group, particularly Erwin and Richard Tyne, who performed most of this work. I'd like to thank the various funding agencies that have supported our work for many, many years. And this is a comment from Bill Vaughan that money won't buy happiness, but it will pay the salaries of a large research staff to study the problem. I only have really a small research staff, so all the things that we could do with ergothioneine, we're not able to do. So we strongly encourage you that the ergothioneine is a fascinating compound to study. Just to end, the journal Feb's Letters in May 2022 published a special issue on ergothioneine, which had 11 papers by leading research groups like Professor Grundemann, who discovered the ergothioneine transporter, Bob Bielman, who I mentioned earlier, a number of Japanese groups that are pioneering the study of ergothioneine in frailty and neurodegeneration, and so on. So if you're interested in ergothioneine, I strongly encourage you to look at this special issue. And let me end by thanking you very much indeed for listening to me today, and please direct any questions to Erwin. Thank you very much.
Okay, so Erwin, if you don't mind taking the acknowledgement to Professor Hallowell, how very interesting his video was, and we really appreciate it. So now we will move on to hearing the seminar by, or the webinar by Professor Eugenio Baroni.
Okay, thank you so much. And first of all, I want to thank the organizers of these seminars, particular Professor Sasso and Professor Butterfield, who invited me for giving this talk about the role of insulin signaling in Alzheimer's disease, and to show you some of our last data about brain insulin resistance development in AD. So for those of you who are not familiar with, Alzheimer's disease is one of the most common forms of dementia among the elderly, and the number of people suffering from AD is estimated to reach around 140 million by 2050. Moreover, we know that almost every 3 seconds, someone in the world develops dementia, and that the cost for taking care of these subjects is estimated to reach $2.8 trillion by 2030. These are numbers, but these numbers tell us that AD today is not only a medical issue, but is also a huge economical and social problem. So we need to put all our efforts to try to find a solution to delay or to stop these catastrophic diseases. From a neuropathological point of view, Alzheimer's disease is characterized by the accumulation of toxic protein aggregates. The most common are the beta amyloids into senile plaques, or the hyperphosphorylated tau protein into neurofibrillary tangles. And the accumulation of these protein aggregates promotes synapses loss, an increase of inflammatory response, an increase of oxidative stress. And during the last years, also a key role for the impairment of brain metabolism, as well as the impairment of insulin signaling in the brain, has been proposed to have a role. The problem is that the clinical symptoms of AD starts several years later than the accumulation of the neuropathological hallmarks. You can see here the violet curve, which refers to memory functions that start to be impaired several years later than the accumulation of beta amyloid or hyperphosphorylated tau, meaning that we need to focus on these early stages to try to find a way to stop the disease. Quite recently, it has been proposed by Fernanda De Felice and her group that insulin resistance is among the components of the allostatic load in Alzheimer's disease, and the concept of allostasis relying with the chronic stress. In other words, we know that our stress are able to respond to stressors, but when these stressors become chronic, our cells do not respond anymore in a proper way. So the accumulation of these stressors represent an allostatic load, and you can see that allostatic load is composed by biological factors, social factors, lifestyle factors, and health outcomes, among which insulin resistance and pathologies associated with insulin resistance, like diabetes, hypertension, or obesity. Indeed, this close connection between diabetes and AD was proposed several years ago, and it was interesting to see that for a long time, brain has been considered an insulin-insensitive organ because insulin does not control glucose uptake within our brain. Today, we know very well that insulin plays a pivotal role in regulating brain functions, either peripheral actions or central actions, including, for example, the regulation of fooding behavior or the cognitive learning functions, which we know to be affected during the development of AD. Insulin was discovered in 1921, but the interest in its role in the brain started around at the beginning of 1990 years, particularly in this paper published in 1995 by Dr. Hoyer. It was proposed that the changes occurring in the brain following the administration of diabetogenic compound like streptozotocin closely leads to metabolic abnormalities in oxidative glucose metabolism that resemble to those found in Alzheimer's disease. So it started to be evident that from a molecular point of view, what happened in the brain in terms of insulin signaling is similar to what happened in peripheral tissues during the development of diabetes. Indeed, we know that diabetes and AD share some pathological features, including increased inflammation, oxidative stress, and the impairment of insulin signaling, and that protein of insulin signaling regulates different processes in different cells. For example, in neurons, activation of insulin signaling regulates long-term potentiation and long-term depression, which are critical processes associated with cognitive and learning functions, while in glial cells, insulin signaling, for example, regulates inflammatory processes. Since we are biochemists, we are interested in characterizing the molecular alterations of a specific pathway and to provide you with some details on how insulin signaling works, because I think that it would be helpful for understanding the next slides. We know that insulin normally binds to the insulin receptor. It promotes the autophosphorylation of the insulin receptor on tyrosine residues, and in this way, insulin receptor can work as a kinases for the insulin receptor substrates. And once activated, the IRS protein leads to the activation of the PI3K-AKT axis, which is one of the arms of the signaling. In turn, AKT activation promotes, for example, the translocation of GLUT4 to the plasma membrane to increase glucose uptake, promotes the activation of mTOR and inhibits GSK3 beta, which are processes associated with the protein synthesis, autophagy, and stress or cell metabolism. In this pathway, there is a novel player of which our group is focusing a lot of work, and this protein is called biliverdin reductase A. Probably you know BVRA because it's the enzyme responsible for the production of bilirubin, which is also a strong antioxidant within our cells. However, several years ago, Professor Maines in the United States discovered that BVRA is target of the insulin receptor, and today we know quite well that once insulin receptor is activated, it promotes the phosphorylation of BVRA and IRS protein at the same time. Through this phosphorylation, BVRA can work as a kinase, and through this kinase activity, it controls the activation of IRS protein, thus forming this regulatory loop upstream in the pathway. In other words, BVRA avoid that IRS protein gets hyperactivated in response to insulin. Furthermore, downstream in the pathway, BVRA can work as a scaffold protein, favoring the translocation of GLUT4 containing vesicles to the plasma membranes, the AKT-mediated inhibition of GSK3 beta, and as we recently proposed, the AMPK-mediated inhibition of mTOR. And you can see here that these processes are key for the regulation of metabolism, stress response, autophagy, and protein synthesis. Moreover, BVRA can work as a shuttle for a member of the MAP kinase proteins within the nucleus, where these proteins promote the transcription of genes involved in cell stress response. You can well understand that because these multiple role of the protein in the insulin signaling. When BVRA does not work properly, we can observe an impairment of insulin signaling, and we are interested in understanding whether the impairment of BVRA affects insulin signaling, thus leading to an impairment, thus contributing to the impairment of cognitive learning functions observed during the progression of Alzheimer's disease. Just to provide you with some molecular details, how insulin resistance develops. This is something common to the brain and to the peripheral issues. Indeed, we know that increased oxidative stress or inflammatory processes leads to the activation of stress-induced kinases that are responsible for the phosphorylation of RS protein on inhibitory sites, which are serine residues. And when this kind of phosphorylation occurs, we can observe the uncoupling between the insulin receptor and the RS proteins. That means that even if insulin can still bind to the receptor, the signaling downstream from RS1 cannot be activated anymore. This phenomenon is called insulin resistance and is associated with reduced glucose uptake, impaired glucose metabolism, and a failure in energy metabolism. These results come from a seminal paper in the field published in 2012, in which it was shown that an accumulation of inhibited RS1 occurs in the brain of Alzheimer's disease, and is also evident in MCI post-mortem brain. But mostly important, to my opinion, it was the fact that they found a very strong and negative association between the levels of inhibited RS1 and the worsening of cognitive learning functions in these subjects. These associations were even stronger than those observed with canonical hallmarks like A-beta or tau phosphorylation, meaning that insulin signaling plays a key role in the regulation of cognitive learning function. What about BVRA? BVRA is widely expressed in our brain, particularly in areas like hippocampus or cortex, and it shares a similar expression pattern with a member of the insulin signaling cascade. Moreover, we already demonstrated that is enough to reduce BVRA levels to observe an impairment of the long-term potentiation, suggesting that BVRA, in some way, we don't know yet if directly or indirectly, controls the processes associated with the cognitive learning function. The story about the role for BVRA in the development of brain insulin resistance in AD started in 2010, while I was doing a PhD internship in the laboratory of Professor Butterfield, because as you well know, Professor Butterfield is a pioneer in the field of oxidative stress. And at that time, we were interested in understanding the antioxidant properties of BVRA. Indeed, our hypothesis was that BVRA may be impaired by oxidative stress, so this would lead to reduction of bilirubin and an impairment of antioxidant properties of our cells. What we found in Alzheimer's disease post-mortem brain is that BVRA is target of oxidative stress modifications like 3-nitrotyrosine, and this is associated with reduced bilirubin production in the hippocampus. So we came up with the hypothesis that oxidative stress impairs BVRA. There is a reduction in bilirubin production, and this would further promote an increase of oxidative stress within our cells that, like in a vicious cycles, further impairs BVRA. Starting from this evidence, and because the role for BVRA in the insulin signaling, we were then interested in understanding whether BVRA impairment may also impact on insulin signaling activation during the progression of AD pathology. Indeed, other than increased 3-nitrotyrosine, we also observed reduced tyrosine phosphorylation in AD postmortem brain, suggesting that BVRA activation is impaired in AD. And as I told you before, this kind of phosphorylation is needed for the proper functioning of BVRA. So to test this hypothesis, we performed a longitudinal study by using the triple transgenic mouse model for AD, in which we evaluated the changes of oxidative stress and insulin signaling activation at different times from 3 to 18 months of age. And you can see here that we observed an increase of oxidative stress starting at 6 months and persisting till 18 months in triple transgenic mice. And when we look at BVRA, we found that BVRA activation was significantly reduced at 6 months and persisted with this reduction in older animals. Interestingly, when we look at the RS1 activation, we found that RS1 gets first hyperactivated in agreement with the role for BVRA in this regulatory loop upstream in the pathway, and only later RS1 gets inhibited. So we identified these two phases, an hyperactivation phase and an insulin resistance phase. And to my opinion, this hyperactivation phase is the more interesting because probably is the phase in which we can still act to prevent the development of brain insulin resistance. These results were also confirmed in a knockout mouse model for BVRA, thanks to a collaboration with Dr. Paul at the Johns Hopkins University, who generated this mouse model. And this is part of the work of our postdoc, Dr. Lanzillotta. And you can see here that knockout mice for BVRA are characterized by increased RS1 inhibition, no changes for AKT, and a reduction of GSK3 beta serine and phosphorylation in agreement with the scaffold function of BVRA at this point of the insulin signaling cascade. We also looked for the hyperactivation phase in these knockout mice, and we found that RS1 is significantly hyperactivated in mouse at 15 postnatal days. Moreover, we focused our attention on mTOR because we were looking for a kinases responsible for the inhibition of RS1. Indeed, mTOR is among the kinases known to be involved in insulin resistance, and it was also found to be hyperactivated during the progression of Alzheimer's disease. Indeed, you can see here that mTOR is hyperactivated already at 15 postnatal days in these mice, and probably is responsible for the inhibition of RS1. And in vitro experiments in which we treated cells lacking BVRA with insulin, demonstrated that the cells are characterized by an increase of RS1 inhibition, while if we treat these cells with rapamycin, a well-known inhibitor for mTOR, the observed increase of RS1 inhibition is not there anymore, suggesting that mTOR is a critical kinases responsible for RS1 inhibition and thus brain insulin resistance development when BVRA is not there. However, we were also interested in understanding the link with oxidative stress because several papers suggest a tight association between brain insulin resistance and increased oxidative stress, particularly at the levels of the mitochondria. And thanks to the support of Professor Elena Forte here at Sapienza University, we were able to study the mitochondrial activity in mitochondria isolated from hippocampal samples of knockout mice for BVRA. Basically, what we found is that knockout mice for BVRA show increased oxidative stress levels in the hippocampus. And when we look at the mitochondrial complexes, we found a consistent impairment of complex 1 and complex 2, suggesting that BVRA in some way, whether directly or through the activation of insulin signaling, impact on mitochondrial activity, which is consistently impaired. These mice show also an impairment of cognitive functions evaluated through different cognitive tests like the Morris-Water maze, the novel object recognition, and the Y maze, meaning that the impairment of insulin signaling and the increase of oxidative stress also affects cognitive learning functions in this model. To strengthen our hypothesis, we performed also in vitro experiments. Here we have the results from an experiment in which we treated cells, control cells, and cells lacking BVRA with insulin, and we evaluated the activation of the insulin signaling at different times. Also in these neuronal cells, we were able to identify this early hyperactivation phase for IRS1 after 30 minutes since insulin administration, while after 2 hours, we observed an increase of IRS1 inhibition, meaning that when BVRA is not there, cells are more prone to develop brain insulin resistance in this case. Interestingly, if you look at AKT, you see that AKT fails to be activated in cells lacking BVRA, suggesting that BVRA is really a key player regulating the activation of insulin signaling. We also looked at the mitochondrial activity, and through the CORs, we were able to evaluate changes of glycolysis and mitochondrial activity in control cells. And you can see here that, as expected, insulin leads to an increase of glycolysis as well as mitochondrial energy production, while in cells lacking BVRA, no changes for the glycolysis or the mitochondrial energy production are observed in response to insulin. Rather, what we found is that insulin leads to a significant increase of mitochondrial oxidative stress in cells lacking BVRA, and these results closely resemble to what we observed in knockout mouse model, in the triple transgenic mouse model, and also in postmortem AD brain. So which are the implications for Alzheimer's disease? Since we were interested in the role for oxidative stress, because our initial hypothesis was that oxidative stress impairs BVRA, and this is responsible for the impairment of insulin signaling cascade, we focused on the effects mediated by oxidative stress in a neuronal cell model. Here we treated our cells with hydrogen peroxide to mimic increased oxidative stress, and we found reduced BVRA tyrosine phosphorylation, meaning reduced BVRA activation, along with an increase of RS1 inhibition. Suggesting that increased oxidative stress impairs BVRA and favors the development of insulin resistance. Then we focused on the AKT-GSK3 beta axis, because this axis, this part of the insulin signaling is quite interesting, since it is also activated as a neuroprotective mechanism, again increased oxidative stress. So we evaluated changes of GSK3 beta serine 9 phosphorylation in cells treated with hydrogen peroxide, and you can see that GSK3 beta serine 9 phosphorylation is increased in control cells treated with hydrogen peroxide, while this is completely prevented in cells lacking BVRA, suggesting that loss of BVRA impairs the ability of AKT to inhibit GSK3 beta. We provided also an explanation for these evidences by showing that the physical interaction between AKT and GSK3 beta is increased in control cells, while it is completely prevented in cells lacking BVRA. We also look at tau phosphorylation, because GSK3 beta is one of the kinases associated with increased tau phosphorylation in AD. And you can see here that tyrosine phosphorylation at the serine 404, which is a target for GSK3 beta activity, is increased in cells lacking BVRA and treated with hydrogen peroxide, meaning that oxidative stress impairs BVRA. This would affect the activation of the insulin signaling, particularly at the levels of the AKT-GSK3 beta axis, which then results in an increased tau phosphorylation. Results collected in vitro were also strengthened in by analysis performed in post-mortem human samples, thanks to the collaboration with Professor Butterfield. And we know that in MCI, this early stage of AD development, reduced BVRA protein levels were associated with reduced AKT-GSK3 beta complex formation, along with reduced GSK3 beta serine 9 phosphorylation and increased tau serine 404 phosphorylation. Last but not least, we focused our attention on autophagy, because autophagy is one of the main mechanisms through which our cells clear toxic aggregate. And we found in knockout mice for BVRA that loss of BVRA impairs the physiological activity of MPK in mediating mTOR inhibition. In other words, loss of BVRA leads to reduced MPK protein levels, as you can see here, and the loss of MPK leads to mTOR hyperactivation, which then results in the block of the autophagic flux. At the end, we observed an increase of oxidative stress levels. So we believe that the loss of BVRA is critical to control several key functions within our brain cells, and in particular of functions normally associated with the development of neurodegeneration. Finally, we also tried to recover BVRA activation or to prevent its impairment. And these results are are shown in this paper. I don't have time to go through these results, but what I can tell you is that we were able to show that if BVRA functions are saved, are preserved, we do not observe the development of brain insulin resistance, confirming our initial hypothesis that probably if we act on this hyperactivation phase, we are able to delay the development of brain insulin resistance and the accumulation of AD-like pathology. So to conclude, we know that BVRA is an essential player of the insulin signaling, and that loss of BVRA promotes insulin resistance development, along with an impairment of cell redox metabolism, which is critical for our brain cells. Moreover, oxidative stress promotes BVRA impairment and the development of insulin resistance early in AD, and that the impairment of BVRA alters the functions of the key proteins like GSK3 beta and mTOR, known to have a role in the development of AD neuropathology. Finally, I want to thank all the people of my group, particularly my outstanding colleagues, Marzia Pierluigi, Fabio Di Domenico, and Antonella Tramutola, and all our students, and of course, Professor Butterfield, who was always with us during these years, and all our external collaborators, and all of you for your attention. Thank you so much.
Okay. Thank you very much, Eugenio. That was fascinating summary of a lot of work. And we'll hopefully have questions here at the end. Okay, so now I ask Professor St. Clair to share her slides and begin her presentation.
Thank you. Could you see my slide now?
Yes, we can.
Thank you. Well, first of all, I'd like to thank Professor Butterfield for the opportunity to tell you about a project that I started over 2 decades ago. So as you can guess, today I can only focus to highlight major study that lead to the current conclusion that the primary antioxidant enzyme manganese containing superoxide dismutase, which sit in the mitochondria, has a dual role in cancer. As a redox active antioxidant, it protects normal tissue against toxic side effects of cancer therapy, but it can also act as an anti-cancer agent. And I will briefly illustrate how this knowledge can be translated into clinical application. Like many of us, my work is built on the shoulder of giants before me. In this project, the two giants were Dr. Erwin Friedrich, who discovered superoxide dismutase in 1968 with his graduate student Joe McCord, and the Larry Oberle, who published a review in superoxide dismutase and cancer, which have become a citation classic in cancer research with his postdoc, Gary Bettner. So why manganese superoxide dismutase? Since, as you see in the Professor Hallowell shown, it is only one of the antioxidant enzymes in the series of antioxidants that work in concert. As shown by Professor Friedrich, superoxide catalyze dismutase, catalyze the dismutation of superoxide to hydrogen peroxide and molecular oxygen with a diffusion rate. And this is really important when it comes to the mitochondria, because mitochondrial electron transport chain produce more than 2 micromole superoxide per second. without manganese superoxide dismutase in the mitochondria, the concentration of superoxide in the mitochondria will be greater than that of molecular oxygen within few seconds. So it should not be too surprising that all aerobic organisms could not live without manganese superoxide dismutase. And this is probably best illustrated in the manganese superoxide dismutase knockout mice generated by Dr. Ting Wong at Stanford University, where they show that the mouse knockout mouse born smaller than the wild type counterparts, and they live in aerobic environment for 2 or 3 weeks, followed by die from massive multi-organ failure, especially massive cardiac injury. Around the same time, my first graduate student, Dr. Yen, generated transgenic mice that overexpress manganese superoxide dismutase in the heart and showed that it actually protects the cardiac injury against doxorubicin, which is a redox cycling drug, generates superoxide radical in the mitochondria. So this study is actually the first study to demonstrate that in this superoxide generated in the mitochondria by anti-cancer drugs such as doxorubicin can cause significant cardiac injury. But the good news is overexpression of manganese superoxide dismutase can reduce the injury. However, people without cancer will not likely to be exposed to anti-cancer drugs such as doxorubicin. So they are quite concerned about if we are trying to overexpress superoxide dismutase to protect side effects of anti-cancer drugs, it may also reduce the efficacy of anti-cancer drug to kill cancer because, as you may know, more than 50% of anti-cancer drugs FDA approved and radiation generates free radical at a mean to kill cancer. Now, that's a valid concern. So to test how that valid concerns is valid, we overexpress superoxide dismutase in the aggressive cancer cell. And then Dr. Yolano implanted into syngeneic mice, that means those mice have normal immune system as well. The result that he showed is not quite what we expected, as demonstrated by TCD50, which is the radiation dose that required to control 50% of radiated tumor. As you can see here, the higher superoxide dismutase activity, the lower radiation dose that required to cure the cancer. So yes, overexpression of manganese superoxide reduce the tumor control radiation doses. This is another really important and good news because it does suggest that perhaps superoxide dismutase have a opposite role in normal and cancer tissue, and perhaps we can use overexpress manganese superoxide dismutase to get this beneficial effect. To search for a way to induce the expression of manganese superoxide dismutase, Dr. Steven Wan, a postdoc in the lab, cloned the human manganese superoxide dismutase. And he showed that it is a single copy gene, which is regulated by a highly unusual GC-rich promoter. Now, this promoter not only binding site for housekeeping protein, but also that of a tumor suppressor gene and oncogene. Dr. Young Su, another postdoc, identified that the strongest enhancer element of the gene is in the second intron. It is an NF-kappa B binding site. And this binding site, NF-kappa B enhancer, can be regulated long distance by tumor suppressor gene or oncogene, depending on what they present at the time. So this together, this result suggests to us that expression of superoxide dismutase in cancer may change depending on the state of the cancer. To test it, Dr. Sanjit Da, another postdoc, took this in human manganese SOD promoter and enhancer to drive the expression of reporter gene, in this case luciferase. He then made transgenic mice that overexpress this construct on the skin, and then applied chemical carcinogen to the skin of the animal. With this animal model and method, now we can observe the change in superoxide dismutase non-invasively from normal skin tissue to when the tumor begin to develop and to progression without having to kill the animal. The result shown here is quite interesting. In the animal that receive the chemical carcinogen, even in the normal pathological looking skin, the superoxide dismutase activity already reduced. And this reduction continue until benign tumor occur, which indicate papilloma. However, when this tumor progress to a very aggressive tumor, now the expression of manganese superoxide dismutase hugely increase. This data is quite critical because we know that the product of superoxide dismutase is hydrogen peroxide. So the data in the previous slide showing you that in this at tumor progressive hydrogen peroxide increased in this tumor. So this prediction is actually consistent with the result from Gary Bettner at the University of Iowa, where they directly measure, compare hydrogen peroxide in normal and tumor cells, and found that in the tumor have higher, significantly higher hydrogen peroxide. Together, our result and Gary Bettner result leading to the current recognition that cancer cells are usually under higher oxidative stress than normal cells. This knowledge and concept become very important, as you will see later. That knowledge coupled to the current knowledge of how redox signaling regulates signaling in vivo, that is under reducing condition. Reducing condition regulate physiological signaling. Now, if you increase oxidant stress slightly, it can activate adaptive signaling. But if the oxidizing condition continue to grow, it will begin to activate this signaling. And as I just showed you earlier, now we know that in the normal cell, they are more in the reducing condition compared to the tumor cell. So because of this knowledge, the idea is if we increase oxidative stress at the dose where it's just sufficient to activate adaptive response in normal cell, this same therapeutic dose should activate the death signaling in tumor cell, pushing tumor cell to oxidative stress overdose and death. Because of our study with manganese superoxide dismutase and this concept, we have been working with Dr. Ines Kebali at Duke University, who generate multiple generations of manganese superoxide dismutase mimetic in form of manganese superoxide porphyrin. Two of her porphyrin have entered clinical trial, but I personally like the third generation of her porphyrin that called BMX001, because in addition to these 5 positive charge, it has 4 long hydrophobic chain that allow efficient transport into the mitochondria. So in collaboration, in order to test if this manganese SOD mimetic viscid and redox active antioxidant can act similar to authentic manganese superoxide dismutase in normal and cancer model. To do that, we start with the normal hematopoietic system, focus on hematopoietic stem cell, because these stem cells are precursor of all mature blood cells. And cancer therapy usually depletes a blood cell in cancer patients. To do this, Dr. Yan Min Chao in the lab did the test in vitro and in vivo. In vitro, she isolated bone marrow stem cell, put them in culture, and treat them with vehicle or manganese superoxide dismutase mimetic, and then transplanted into recipient mice. Or she treated the mouse, normal mouse with vehicle or manganese superoxide dismutase mimetic, and then isolate the bone marrow stem cell, transplanted into recipient mice. As positive control, she isolated bone marrow stem cell from either wild type mice or manganese superoxide dismutase transgenic mice and transplanted into the recipient mice again. Her results show that in cell culture, in cell, the percentage manganese superoxide dismutase mimetic increased the frequency of hematopoietic stem cell as shown here. As a dose-dependent manner. In animals that receive the mimetic for 100 days, you can see significant increase in hematopoietic stem cell. And this is consistent with what's found with the manganese superoxide dismutase transgenic mice. So yes, MNP or this mimetic can behave similar to authentic manganese superoxide dismutase in the animal model. For the blood cancer, Dustin Carroll, a graduate student, used myeloid dysplastic syndrome, visit a type of blood disease that eventually progressed into leukemia. Here, Dustin showed that indeed the MNP significantly and efficiently killing the MDS cell. So mechanistically, Dustin showed that MNP, because it is a redox cycling, it can generate hydrogen peroxide in both normal hematopoietic stem cell and leukemia cell. However, because of normal hematopoietic stem cell are under reducing condition, this hydrogen peroxide mainly oxidize KEAP1 that will allow NERV2 to go into the nucleus and transcribe the expression of antioxidants such as catalase, manganese superoxide dismutase. In contrast, this hydrogen peroxide that increase in the leukemic cell under the heightened oxidative stress condition in the leukemia cell activate AP1, which when go into the nucleus, activate transcription of deadly latest genes such as P53 and various caspase. So here MNP behaves similar to manganese superoxide dismutase in that it can efficiently kill cancer cells as well. So together with the normal hematopoietic stem cell, it would appear that MNP, as a redox active antioxidant can protect normal hematopoietic stem cell and kill cancer cell. Now these concepts are not limited to blood cancer. Summarized nicely by Dr. Ines Batani-Hebali in this slide, to combine with what I just shown you. This study shown in experimental therapeutic model, it drives the progress of MNP into clinical trial. One of the clinical trials that we participate is to try in high-grade glioma. In the phase 1 clinical trial, the results suggest that indeed BMX-001 increased survival time by 50% and also protect against side effects of radiation and chemotherapy. This result then pushing the clinical trial into the phase 2, which is the randomized trial of newly diagnosed high-grade glioma treated with concurrent radiation, temozolomide, with and without BMX-001. The recruitment are now complete, so hopefully we will have the result to share with you very soon. What I would like to show you is that the concept is what I want to emphasize. Besides the MNP that I have shown you, another superoxide dismutase mimetic that generated by a different company named Galera they call the compound GC compound. In September of 2021, Kellogg's announced that they announced the result in the pancreatic cancer trial showing that the GC compound improved all the efficacy parameters observed in the overall survival, progression-free survival, local tumor control, time to metastasis, and response to tumor rate. However, in this study, they did not look at the protection in normal tissue. But Kellogg's recently, just earlier this year, announced that they have completed the phase 3 trial of the GC compound for severe oral mucositis in patients that received radiation and chemotherapy for locally advanced head and neck cancer. The trial showed that the GC compound reduced the severe oral mucositis both in the incidence and relative and duration of injury. So together, this study showing that REDOX active antioxidant very exciting potential new therapeutic drug that we could use based on the concept of this compound can kill with what we call two birds with one stone. With this exciting possibility, we are now begun to identify FDA approved drug with this property in hope of expanding the possibility of curing cancer and protect normal tissue at the same time. And I hope that you join us to expand this exciting new field of research. Of course, research is a team effort, as you know. I'm so fortunate that at the University of Kentucky, I have such great students, a postdoc, faculty, collaborator, even supportive administrator constantly throughout our study. And of course, money is really important. We really appreciate the sustained constant support of NIH for our research and various private foundations, especially the James Graham Brown Foundation in Delaware, which allow us to be able to initiate high-risk, high-impact study before NIH agreed to fund us. And thank you so much for your attention. I hope you will expand the field further with us. Thank you.
Okay, you can now unshare your slides. And thank you very much, Professor St. Clair. That was just a fascinating presentation. All right, we can now open these three talks for question and answer. And so let me look through some of the questions here. And we maybe can start with the Hallowell presentation. So a question from Victoria Bunik from Moscow State University. You refer to some trends. This is now directed to you, Erwin. You refer to some trends, but the parameters are measured in some homogeneous samples. Specific locations may have more significant changes. Secondly, slide 23, is it possible to take into account metformin administration in your groups? As many old people take this anti-diabetic that is transported through OCT. So maybe a complication is what she's implying here. All important cations, including ergothioneine or thiamine, could be decreased, especially in some polymorphisms. You have some thoughts on that, Erwin?
Yeah, sure. I'm not sure which the first part is referring to. I mean, if it's referring to the human studies that we did, so in the young healthy volunteers, yes, they are quite a small population. It's a very narrow age group. So these are very young volunteers, and of course we didn't expect to see massive changes in the oxidative damage markers because, you know, I mean, there's really nothing wrong with them. I mean, they have their own endogenous defenses. So, yeah. So we didn't expect to have to see a huge change, but there were some trends that we noted. Yeah.
Now, yeah,
I'm not sure if that answers the question.
well, yeah, but now talk about the other one where other molecules can also
Yes,
be transported by OCT.
Yeah, that's a great question. So we've we ourselves and Grundemann, who's the person that discovered the transporter, actually looked into the specificity of transporter. So there's been many other groups that actually have reported that OCTN1 transports other drugs such as metformin. But we've sort of shown that OCT is actually very specific for ergothioneine. Sorry, it's the other way around. So ergothioneine is a very good substrate for OCTN1, and it's transported at greater than 100 times efficiency, to the point where I think the other group is actually considering renaming the ergothioneine transporter. So we don't think that other drugs actually interfere with the uptake of ergothioneine as such, and if it does, it'll be very minuscule.
Okay.
Yeah.
Thank you. That's very helpful. And then have you looked, let's see, I guess there was another question. We'll maybe ask one more for you, Erwin.
Yeah.
From Antonio Curado. I'm sorry for mispronouncing your name, sir. And the question is, considering that ergothioneine is a redox cycling molecule, most likely it inhibits KEAP1, leading to NRF2 activation. Have you looked at surrogate biomarkers of NRF2 activity in blood treated subjects, of ergothioneine treated subjects?
Yeah, that's a great question. We haven't specifically looked. There are some other groups that actually have reported that ergothioneine is able to inhibit KEAP1 and activate NRF2, and also the downstream genes such as the HO1 and QO1 and so forth. Yes, that's a good point. We haven't actually looked in our human samples, but that could be something that we look into.
And it relates, doesn't it, to Professor St. Clair's talk, right? Because manganese SOD also comes off that, right?
Yes, that's right. Yeah,
that's pretty interesting. Okay, let's see. So some questions for Professor St. Clair. Let's see. I'll start with this bottom one first. Paula Mayacote from Mexico. Is the effect on cancer and normal cells dependent on hydrogen peroxide accumulation, and can it be reverted by catalase?
Very good question. In fact, it depends on hydrogen peroxide to a certain extent. We are focusing on hydrogen peroxide and superoxide because, as you know, location is very important. Depend on where that radical initiate. In the mitochondria, mostly a superoxide. And then the catalase is not in the high amount in the mitochondria. But if your general question is, if catalase can do the same thing, it can remove hydrogen peroxide, but perhaps because it did not have both a reduction and oxidation capacity like superoxide dismutase. It is a two-step oxidation, a reduction step, but the oxidation step is not similar to superoxide. Therefore, my case would be probably moderated, but would not have a capability to kill cancer cell and not protect normal cell. It will protect normal cell for sure.
Yeah. Thank you, Dorette. That's great. There's another question for you from Dr. R.B. Singh from India. Let's see. let's see if I can do it. How cancer cells are formed. He mentions cancer is a disease of genes, but what is the first event with reference to epigenetics? And I'm not quite sure that. Oh, and then he also is asking initially, how are cancer cells formed and how with reference to apoptosis?
Well, that's a couple $1 million question. So let me try to narrow it down first. You are right. Cancer, in the cancer field, we believe that it is a problem of DNA. As you know, this is from the beginning of a century. We know that cancer, for it to completely develop, it requires 2 hits. For example, the model that we are using, such that we use DMBA and TPA. The DMBA will cause a mutation of the Harvey-RAS gene. And yes, it initiates the mutation of the gene, but that mutation itself is not sufficient to lead to the development of cancer. And that's why we need the TPA that generates generate free radical as a promoter. So in that model, you can see that it is the initiator and promoter, with it consistent with what you say that cancer is a disease of the gene. But the disease of the gene itself is not sufficient to develop into full-blown cancer. And as you see, maybe because I did not go into the detail, even in the skin cancer study that we show you, after the cancer initiate, when the superoxide dismutate begin to decline, when the gene, the Harvey-RAS gene already mutate, the enzyme activity reduced. But if pathologists look at them, they will tell us that this is normal skin. So we call that an initiator. But when TPA come in, it begin to cause superoxide, hydrogen peroxide production. Initially, the animal would not be able to immediately wrap up the arrested response. So you can see that the benign tumor, actually the enzymes decrease. But during the progression step, that after the gene have been mutated, right, you see a huge adaptive response of increase in superoxide dismutase. So at that point, a huge response is what we suspect that epigenetic begin to play a role. And of course, epigenetic, in my mind, involve more of a promotion step as opposed to our traditional gene mutation, genetic defect type. But of course, as you know, epigenetic also control the gene and the expression of the gene. So they are related in circle, but the tumor, especially different type of tumor, have its own unique capability. In some tumor, you have p53 mutation, and p53 is the most mutated tumor suppressor gene. But yes, it only mutates in 50% of the cancer, right? And a lot of the, another 50% may have p53 mutated, but you do not get cancer, for example. So it's a little bit more complicated than one step, clean cut, but we are getting there.
Yeah, for sure. Thank you very much, Professor St. Clair. And there's another question related to you, but I will come back to that. So now I want to ask Professor Baroni a question first. And this is from me, but it's not in the chat, but so I'll sort of formulate the question directly from my head. Just thinking about GSK3 beta and BBRA's relationship to that, that was pretty fascinating. I thought that was great. But do you know that GSK3 beta, similar to protein phosphatase 2A, both target tau phosphorylation, right? And both are defective in Alzheimer's disease. And part of that might be because of they're both regulated by a common protein, which is called PIN1.
Mhm.
Are you familiar with PIN1? And because our lab showed PIN1 is much lower level in Alzheimer's brain, and it's oxidized, and its activity is way down. And maybe, do you think there's another step? That's what I'm getting at. That BVRA might even be
Well,
conceivably under PIN1 regulation, because it does regulate dozens and dozens of proteins. What do you think about that?
yes, this is a really interesting model, I would say, because I mean, I think that GSK3 beta is a part of different pathways, intracellular pathways within our cells, and is activated or inhibited. I mean, it is already autonomously activated within our cells and is inhibited by different active intracellular pathways. So
Right.
probably, I think that it depends on the condition we are looking for. So probably PIN1 may have a role, BVRA may have a role through the insulin signaling. For example, I didn't have time today, but the story about the interaction between BVRA and GSK3 beta is, for me, even more interesting because we have additional data showing that they both impact on mitochondrial bioenergetics in a specific way. So I hope that we will publish this data quite soon. And the interesting thing in GSK3 beta is that what we found is the fact that GSK3 beta is hyperactivated during the early stages of AD. As you well known, and as you already also published, the GSK3 beta is mostly inhibited in AD, while if we look at the early stages, probably is there that we see this hyperactivation of GSK3 beta. Of course, GSK3 beta is not the only kinases responsible for tau phosphorylation, but the phosphorylation of GSK3 beta also serves as a primer for other kinases, so contributing to the accumulation of tau phosphorylation in AD. Altogether, maybe these mechanisms, including also LRP1 and the other, the phosphatase A2 and the other kinases associated with tau phosphorylation, contributes to the formation of neurofibrillary tangles. I think that they contribute to the development of these kind of abnormalities.
I kind of anticipated that answer because it is a complex situation for sure. Yeah. But it's interesting how one protein is able to regulate so many others by just changing a serine, let's say proline, serine. So it's a phosphocserine proline. There you go.
Exactly.
In that sequence, or phosphothreonine serine, and just change the proline from cis to trans, change the structure of that protein so much that its activity is tremendously regulated. And that doesn't work in Alzheimer's. Another one of its targets is amyloid precursor protein. And so the two main pathological hallmarks of AD both are regulated in some ways by PIN1, which is
exactly.
Well,
this is very fascinating. So
yeah, for sure. Let me thank you, Eugenio. Let me go back to a question from Victoria Bunick. She's from MSU. I'm not quite sure what that means. Michigan State University, Michigan State University, Montana State University, or some other university. In the United States, it could be any number of things, but that also might mean another country. Anyway, she asked a very interesting question, and this is to you, Dorette. It's about she thanks you for sharing your examples of the different responses of cancer and normal cells to redox sensitive antioxidants. And she argues that she has seen this when treating cells with thiamine diphosphate. This is an essential coenzyme for animals with side effects unknown, but damaging in the same dose for cancer cells. And she was expanding a little bit in a subsequent post and said, this difference in thiamine diphosphate effects in normal and cancer cells is linked to their initially different redox metabolism. So any thoughts about the relationship between thiamine diphosphate and that's a cofactor, if I remember, to pyruvate dehydrogenase and alpha ketoglutarate dehydrogenase. So what do you think of that?
Yes, I think that's an extremely interesting question. And clearly, it's a cofactor of the mitochondrial electron TCA cycle. So in the old day, one used to believe that cancers are glycolysis, right? And then did not derive much of energy from oxidative phosphorylation. Now we know that that is not true. The more aggressive the tumors are, the more they have the ability to use oxidative phosphorylation. So now they are competent for both glycolysis and oxidative phosphorylation.
Right.
However, as we suspected that in those tumors, they already came up their metabolism oxidative. So as you know, even under normal situation, we sit here, our mitochondria leak superoxide, trillion of molecules per second. And just imagine the tumor cells that are carrying it to the full extreme would have more of the superoxide, more leakage of the electron from the mitochondria electron transport chain. So that will build up a significant amount of superoxide, hydrogen peroxide diffusible. So I would think that it is actually not so surprising that you would see the effect in killing the cancer cell. However, what I suggest is, because as I mentioned earlier, I did not have time to show you, we have begun to screen FDA approved drug using the concept of the compound, the agent is redox cycling. Using and they also generate hydrogen peroxide. I would not, so I would suggest that you knock it out or you add it to the tumor cell and then measure hydrogen peroxide directly. You can also treat the normal cell, and this might be another intrinsic metabolism derived way of difference between normal and cancer cell. But the same concept should apply here. Is it exciting? It's a good question.
Yeah, thank you. Good answer too, because very relevant to even Alzheimer's disease is thiamine, and that it leads to this compound DPP. And that's Gary Gibson in Cornell has studied this a lot in Alzheimer's disease, actually coming to our university in January to give a seminar about that. Okay, so Victoria, I hope you got an answer to your question. And Dr. Moscow State University, aha, sorry. That's where she's from, okay. Thank you for that. And then we have another comment from Dr. Singh in India, and says this, and I kind of agree with this, having known Dr. St. Clair for quite a long time. He says, Durrett, you are amazing. Can you try to develop an epigenetics test to diagnose cancer at initiation of its effects on the epigenome? For example, gene methylation or methylation of I'm not quite sure what the other stuff is. So methylation of another aspect. Oh, chromatids. Yeah. Okay, so if you have any thoughts on that, Durrett?
Yes, actually, thank you for the question. This is, of course, the epigenome it's not that new, but just recently become a very, very exciting area of cancer research as well. And in fact, at the University of Kentucky, we have a special program called Center for Cancer and Metabolism, where one of the projects focus on epigenetics, which regulate the metabolism. And in this methylation of the epigenetic, for example, so I come back to what I know again. Even though even the manganese superoxide dismutase gene, I show you that the strongest enhancer element is on the intron. However, the genes in some of the tumor cells really heavy methylated, and but that only occur in the benign tumor, and that's one of the reasons why the gene did not express during the promotion, but they eventually overcome that defect. But yes, epigenetic regulation of the gene during the tumor development have become a very, very exciting major area of research that we actually have a lot of junior faculty working on it.
Okay, thank you. Let's see, I hope I have gotten everybody's questions. Oh yes, here's one more for Irwin, and referring to Professor Hallowell's talk. Does ergothioneine act as a pro-oxidant if we use it at high dosage levels?
Yes, good question. So I think Rafael mentioned that so at physiological pH, the ergothioneine actually exists in a thioane form. So this confers the stability and also prevents the auto-oxidation like other thiols. So we haven't seen any oxidate at high doses. We haven't seen this overt stress. There's been toxicology studies done in rats at very, very high doses, like a few thousand times higher than what we've ever given in any animals, and they're perfectly fine. There's no overt symptoms.
Good to know. And would you recommend? I sort of heard this from Professor Hallowell's talk, but are you recommending that more people incorporate mushrooms into their diets?
Yeah, so I mean, it was a correlation study to show that the people that consume more mushrooms had a lower risk of developing cognitive impairment. But of course, mushrooms contain a huge amount of different compounds.
I understood. Yeah, you're not necessarily ascribing that to ergothioneine, but maybe. Yeah. But nevertheless, the mushrooms themselves somehow seem.
Yes. There's always a.
Yeah, Barry and I once had dinner together at a meeting in I think it was in Indiana maybe, and in there he always liked to get a big steak and a giant plate of mushrooms. And now I understand why he wanted to have the mushrooms there.
Getting his daily dose.
That's get his daily dose, exactly. Okay.
Definitely something that I take notice. Because
me too, Durrett.
The mushroom to begin with. I also learned from Eugenia that I better check my insulin because it really related to
Yeah, I think that really all three speakers really tied the theme together so well about redox metabolism on the one hand and pharmacology on the other. That was really great, all of you. And I hope that this inspires all the attendees to think along the lines of what you were instructed in today by these wonderful speakers. And I, for one, am very, very grateful to each of you for your outstanding science. And Erwin, you are, you have a wonderful career ahead of you by pursuing these sorts of studies. At some point, Hallowell is going to have to retire. But somebody's going to have to take his place. And I'm saying that out of something close to my career as well. And somebody like Eugenio and his colleagues there, Marzia Pierluigi and Fabio Di Domenico and Antonella Tramutula, and some of the new postdocs in the group. I mean, these are the next generation of people who are going to carry redox biology into the future. And so we're really proud of them. Okay. I think with that, because Professor Sasso had to run to another appointment in Rome, I just want to thank him and the organizers of this symposium for asking me to put it together. And I must say, this turned out to be really fascinating. And I thank all the attendees for taking part and listening. It's wonderful to note how many different countries of the world are represented by people who signed in. And this is just one of the positive things, if there are any, but this is one out of COVID, has originated this Zoom technology that has just revolutionized the way people can communicate from all over the place at one time. And it's simply fascinating. And anyway, I thank you all for tuning in, and I hope you found this as exciting and as interesting as I have. And I thank all the speakers again for just tremendous science they're doing, just tremendous science. So thank you again. And now I guess, Marco, I will let you sort of close out the Zoom for us. So all the best to each of you in your respective time zones. And I hope everything goes well. And Eugenio, say hello to everybody over there in Rome for me.
Of course. Thank you so much.
Okay. Take care, everyone. Bye-bye.
Thank you.
Bye.
Bye. Bye to everybody. Thank you.